I'm an associate professor of nutritional biochemistry in the Food Science and Human Nutrition Department at the University of Florida. My research focuses on the molecular mechanisms of iron transport. Basically, we're trying to figure out how iron, the essential nutrient, or essential metal in this case, is taken up by cells. I'm trying to figure this out because disorders of iron metabolism are some of the most common disorders affecting humans. Iron deficiency, for example, affects over 2 billion people in the world. These are estimates from the World Health Organization. And when iron deficiency is severe, it can lead to iron deficiency anemia. On the other hand, there are several disorders of iron overload which are not uncommon, and these include the genetic disorder hereditary hemochromatosis, and then a red blood cell disorder called thalassemia major. One of the problems that my research focuses on is how does iron get taken up by the liver? And to put this into perspective, the liver is where we store excess iron. Our data indicate that the protein that the liver uses is a protein called ZIP14. One of the ways we show that is we have a variety of experimental models of this iron overload disease, hemochromatosis, but without ZIP14. And for example, in animals that lack the ZIP14 protein, they have a lot of iron in their body, but it isn't taken up by the liver. Instead, it's taken up by other tissues, namely the kidney and the heart and the lung and so forth. We're also looking at how iron is taken up and crosses the placenta. In fact, iron transport across the placenta is an amazing process. In that 9 months of pregnancy, an enormous amount of iron is transferred from the mother to the developing fetus. One of the ways that we show protein can transport iron is we use cells in culture. And what we do with these cells is we can get them to overexpress our protein of interest, or we can do experiments where we can take the cells and suppress the expression of this particular protein. In both those situations, then, we can take and give the cells iron and see how much iron they take up when the protein is overexpressed, and see how much iron is taken up when the protein expression is suppressed. So if it takes up more iron when it's expressed and less when it's suppressed, that is pretty strong evidence that, at least in cells, this protein functions in iron transport.